At the chemical synapse, in order to make that transition from electrical signal to chemical signal back to electrical signal, we're going to need to release neurotransmitters. These are chemical signals released from neurotransmitters released from neurons at the synapse. Note, we call some neurotransmitters excitatory or inhibitory, but the actual effect of neurotransmitters are going to depend on the receptor. Some neurotransmitters are excitatory in some parts of the body and inhibitory at other parts of the body because different postsynaptic cells have different receptors for the same neurotransmitter. Excitatory neurotransmitters are typically going to cause an influx of sodium. That's going to lead to a depolarization. Inhibitory neurotransmitters are going to cause either an influx of calcium, which is a negatively charged ion, which is going to make the cell more negative, or an efflux of potassium, which is going to make the cell, again, more negative, or what we call hyperpolarization. We also have some neurotransmitters that we call neuromodulators. And neuromodulators, instead of directly causing an EPSP or an IPSP, an excitatory postsynaptic potential or an inhibitory postsynaptic potential, it instead is going to affect the overall behavior of that neuron. Our neurotransmitter receptors, again, this is what actually causes the effect of the neurotransmitter. We have as either ionotropic receptors or metabotropic receptors. Ionotropic receptors, or channel-linked receptors, are chemically gated ion channels, or what are called ligand-gated ion channels. The effects of them are relatively quick and brief because all they're doing is opening up and letting ions flow through. And once an ion channel opens up, the results are very, very rapid. So excitatory receptors are typically going to be channels for sodium. Sodium is going to want to flow into the cell. Inhibitory receptors are going to typically be channels for chloride. Chloride is an anion, a negatively charged ion. So when those open up, it's going to make the cell more negative or make it harder for the cell to become more positive. Also true if we open up channels for potassium. Metabotropic or G protein linked receptors, these are not chemically gated ion channels. Instead, they are what are called G protein linked receptors. G protein linked receptors work by after the neurotransmitter binds to the receptor, it activates what's called an associated G protein. Then that associated G protein goes on to have effects in the cell through the production of what are called second messengers. Often it will activate an enzyme called adenylate cyclase, which produces a second messenger called cyclic AMP that can do all sorts of things like open or close ion channels to, again, allow for graded potentials to form. It can activate what are called kinase enzymes, which are enzymes that add phosphate groups to other proteins, some of which are also kinases. So you can have kinases, and these cause what are called signaling cascades inside of the cell that can cause all sorts of things like turning on or off different genes and making different proteins be made. It can also change the overall behavior of channel proteins, making them more or less likely to open and making the cell more or less likely to fire an action potential. But it's not through directly opening up an ion channel. It's through activating a G protein, which activates another protein, which may activate another protein, which then opens a gate. So these tend to be slower responses, but they can be more complicated, produce more effects inside of the cell. So different types of neurotransmitters. Our body makes a lot of different ones. One of the first identified and best understood neurotransmitters is a neurotransmitter called acetylcholine. Acetylcholine was in part discovered due to the effects of the vagus nerve on heart rate. So when the vagus nerve is stimulated, it actually slows down the heart. This is due to the release of acetylcholine. It was eventually discovered. So this was actually how neurotransmitters were originally discovered. They found that if they stimulated the vagus nerve enough, even if it was not directly connected to a heart, if they took the resulting juices and put that onto a heart, the heart would slow down. So they knew that there was some sort of chemical signal. So this acetylcholine is also the neurotransmitter that is released at neuromuscular junctions. Acetylcholine is going to be made by adding acetic acid to choline through the enzyme choline acetyltransferase. We also have an enzyme called acetylcholine esterase, which is going to break down acetylcholine. Acetylcholine, you might be confused because acetylcholine causes muscles to contract. So why would it slow down heart rate? Well, at skeletal muscle cells, we have what are called nicotinic acetylcholine receptors because they respond strongly to the chemical nicotine. And these are ionotropic receptors that act as sodium channels, and they are excitatory. However, there's another type of acetylcholine receptor that responds to a chemical called muscarine, which is a poison found in certain types of mushroom. So these are muscarinic acetylcholine receptors. These are metabotropic receptors that we find in cardiac cells and other targets of the parasympathetic nervous system, and it's actually inhibitory at these receptors. So depending on receptor, acetylcholine can either cause skeletal muscles to contract, or it can make heart muscles slow down. We have a class of neurotransmitters called biogenic amines. These are also called monoamines. And we divide these into catecholamines and indolamines. Catecholamines include norepinephrine, epinephrine, and dopamine, and they're made from the amino acid tyrosine. So they're called amines because they're made from an amino acid. We also have the indolamines, which are serotonin, which is made from tryptophan, and histamine, which is made from histidine. These are typically going to be more neuromodulators, regulate how the cell behaves, and it's widely used in the brain in order to regulate emotional states and biological clocks, and have these really powerful, wide-ranging effects on overall behavior of the brain. The signal termination for monoamines is typically going to be regulated by what are called reuptake pumps and monoamine oxidases, which are enzymes that break down our biogenic amines. Imbalances in our amines are often associated with mental illness, and drugs to treat mental illness are often going to regulate signaling. For example, we have SSRIs, or selective serotonin reuptake inhibitors. Most antidepressants fall in this category of SSRIs. So here we have a neuron releasing serotonin. Normally that serotonin would be sucked back up by a reuptake pump, but here we have an SSRI blocking the reabsorption of serotonin, so serotonin builds up in the synapse and we get more signaling. It's like we had more serotonin. We're not actually making more serotonin, but it sticks around in the synapse for longer and has more of an effect. We also have monoamine oxidase inhibitors, MAOIs, which are going to be drugs that inhibit the monoamine oxidase, and so again, the monoamine stays in the synapse for longer and has a stronger effect. Then we have amino acid neurotransmitters. These are neurotransmitters that aren't made from an amino acid, they just are an amino acid. So this is going to include glutamate, GABA, and glycine. So glutamate, this is the primary excitatory neurotransmitter in the central nervous system. It's going to bind to receptors called AMPA, AMPA, and MNDA receptors. The AMPA receptor is going to be a sodium channel, so the opening up of AMPA receptors is going to depolarize the cell, so be excitatory. If a cell has already been depolarized, then NMDA receptors will respond to glutamate, and this also allows calcium to enter. And calcium, once it gets into the cell, is going to trigger something called potentiation, which actually strengthens this synapse. And so when a synapse gets used repeatedly in rapid succession, it makes the synapse stronger and increases the connections. This is the basis of something called Hebbian plasticity, which is the basis of learning in the brain. We have another amino acid neurotransmitter called GABA, or gamma aminobutyric acid. This is going to be the primary inhibitory neurotransmitter in the brain, and it's going to bind to what are called GABA receptors. And GABA receptors are chloride channels. Again, chloride is negatively charged. It's at a higher concentration outside of the cell. So when chloride channels open, chloride tends to flow into the cell, making it more negative, less likely to fire an action potential. And then we have glycine. Glycine is going to be an inhibitory neurotransmitter that we see in the spinal cord, and it's going to regulate the behavior of motor neurons. It is actually the target of tetanus toxin. Tetanus toxin is a toxin produced by a bacterium called Clostridium tetani. This is a relative of Clostridium botulinum, the bacterium that causes botulism, but it has the opposite effect. Whereas botulinum toxin prevents the release of acetylcholine, and so all of the muscles stay relaxed. Tetanus toxin prevents glycine from working, and so it's going to prevent these inhibitory motor neurons from inhibiting inhibitory interneurons from inhibiting motor neurons. And this is going to lead to a disease state called tetanus. This is when all of the muscles tighten up, contract, and this can actually produce muscle contractions so strong that they can actually break bones and be fatal, primarily through asphyxiation. Then we have peptide neurotransmitters. So what are called neuropeptides? So instead of being one amino acid or made from an amino acid, these are short strings of amino acids, and they have a lot of different effects inside of the brain and throughout the body. They are going to act on metabotropic receptors, so not receptors that act as ion channels, and they tend to be neuromodulators. So substance P, this is going to be involved in sending pain signals. In contrast, we have endorphins. These are going to be released in the brain, and they are going to act as natural opiates. They actually reduce pain perception. And also we have this whole gut-brain peptide. So we have what's called the enteric brain. It's not actually the brain, but it's a lot of neurons around the digestive tract. And this is actually the second highest concentration of neurons aside from the brain. That's going to play a lot of roles in regulating digestion. So we have somatostatin and cholecytokinin, which is going to play a role in regulating this enteric brain, which regulates digestion. Then we have these gasotransmitters. So some gases actually act as neurotransmitters. One of those is nitric oxide. Nitric oxide is going to bind to these G protein coupled receptors in the brain. And one advantage of these gasotransmitters is that they are lipid soluble. And so they just flow out of the cell, and they can trigger all sorts of nearby cells because they just diffuse away from where they're made. That does mean that they have to be synthesized on demand because there's no way of actually trapping it inside of the cell, because you do that with membranes. And if you're lipid soluble, you diffuse through a membrane. But nitric oxide is actually involved in learning and memory formation, and it's also going to be involved in smooth muscle relaxation in the intestine and blood vessels. This is going to regulate blood flow in the heart. This is also the basis of how the drug Viagra works, through regulating nitric oxide signaling. So the release of neurotransmitters is going to cause a postsynaptic potential. So that's why we call it a postsynaptic potential, because it's going to be a graded potential that forms in the postsynaptic neuron, the neuron after the synapse. And our postsynaptic potentials can be excitatory or inhibitory, EPSPs or IPSPs. Excitatory postsynaptic potentials are going to involve the influx of sodium to cause a depolarization, moves the neuron toward threshold, less negative, more likely to fire an action potential. Inhibitory postsynaptic potentials are instead going to make the membrane more permeable to either potassium or chloride. If chloride channels open, chloride flows out, making the cell more negative. If calcium channels open, chloride moves into the cell, making the cell more negative. That's going to move the neuron farther away from threshold and less likely to fire an action potential. Our EPSPs and IPSPs get added together in a process called summation. A single EPSP does not produce enough depolarization to move the cell all the way to threshold. However, if we have a bunch of EPSPs, they can summate or get added together in order to move the neuron towards threshold. Now, IPSPs can also summate, so we can get EPSPs and IPSPs all adding together, and if the EPSPs predominate over the IPSPs, then we're going to reach threshold and we're going to fire an action potential. We have two ways of summating, temporal and spatial. In a temporal summation, this is going to be based off of the frequency of the release of neurotransmitter. So if we get neurotransmitter released infrequently, the EPSP is going to degrade before we get another EPSP, and we're not going to reach threshold. Remember, PSPs are graded potentials, and graded potentials degrade quickly. So this is like a neuron every once in a while going, fire an action potential, fire an action potential. However, we can also have that same neuron fire a bunch of action potentials rapidly, causing multiple releases of neurotransmitter, each of which depolarize the cell, and these graded potentials get added together or summated. So this is like this presynaptic neuron, instead of going fire, fire, going fire, 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 fire. And that's much more motivating. That's going to be much more likely to get the postsynaptic neuron to fire an action potential. We can also have spatial summation, where instead of one presynaptic neuron saying fire, we have multiple presynaptic neurons all saying fire. And those signals can travel down the axon and build up inside of the soma and eventually reach the axon hillock. And if we have enough of them, then that's going to depolarize the cell to threshold, and it's going to fire an action potential. So this is like a lot of people shouting, fire, shoot off an action potential at the same time. However, with spatial summation, some neurons may be producing ESPSs, and others may be producing IPSPs. EPSPs and IPSPs. Sorry. So in this case, it's like some neurons saying fire and other neurons saying don't fire. And depending on which one has more people and how loud they are, then that might cause the cell to fire or not fire, depending on the ratio between that signal.